Hello students, welcome back. This time around we're going to follow up on our discussion previously on muscle physiology. But previously our discussion on muscle physiology was looking at the end effects at the muscle level. How did we end up getting a contraction to occur, shortening of the cell? If you recall, that was a series of events that began with myosin head group attaching at the active binding site on the active molecule, pivoting, detachment when the ATP molecule bound, cocking of the head group, and then again we had the, attachment, pivoting, and this cyclical event occurred. But remember what triggered the first one, what allowed the first one to occur. Remember that the myosin head group was already cocked, but the myosin head group at rest did not bind to the active molecule because the tropomyosin molecule was blocking the binding site on the active molecule. Now remember what allowed for the tropomyosin to move out of the way was the efflux of calcium being brought into the cell. And that calcium bound to the TNC component, troponin. And when it did that, it caused conformational change, caused troponin to rise up, tropomyosin to slide down, and expose that binding site so that we could see the myosin head group bind. Remember all those events? Now the question is, what set the table to encourage the efflux of calcium inward? And those are a set of electrical events. Now, when we're talking about electricity, we're talking about electrical potential at the cell level. Remember from your nervous system discussion, we have something called resting membrane potential. It's going to come back to play here with the muscles as well. Remember that at rest, there is a net charge across that plasma membrane. In this case, it's called a sarcolemma. Every cell has it, but only a few cells take advantage of it, such as the nerves and the muscles. But remember in the extracellular space that it's going to be, on the outside of the cell, is going to be positive in comparison to the inside of the cell. Part of what makes that difference is going to be the differing concentrations of ions on either side of the cell. Notice from this depiction, sodium is much higher on the outside of the cell than it is on the inside at rest. Potassium is much higher on the inside of the cell than it is on the outside. Now, if you were starting to just add up the concentrations of sodium and potassium on either side, you would not see this great difference in resting membrane potential. What makes the difference, remember, is going to be these A's over here. These A's stands for anionic proteins, negatively charged anionic proteins that are non-permeable, that are only found inside the cell. So the inside of the cell is negative, mostly because of those negatively charged anionic proteins. Let me ask you a question though, is there a potential here to do work, electrical work, considering that these ionic charges will create an electrical change? Another way to ask that, if I were to have a channel right here, and it was a sodium specific channel, and I could somehow trigger that gate channel to open, would sodium move? Which way would sodium move? Would sodium move out or would sodium move in? I'm hoping you're telling me by this point in the lecture, in this course, that sodium would move in because it's a greater concentration on the outside than there is here on the inside. And that it would move based on simple diffusion. Now, sodium itself would move, but also coming with it's going to be that charge associated with sodium. Now, remember the inside of this cell is negative. If I bring positive charged things inside this cell, is it going to make it less negative or more negative? Hoping you're telling me it's going to make it less negative and bring it closer to zero. That process is known as depolarization. Now, let's back up here now. Since we know at rest we've got a potential, let's see the events that go on in the excitation events of a muscle contraction. Here we have a major motor neuron that's heavily myelinated. Notice as it gets closer to the muscle, it will branch out into its axonal termini. Here's a blow up of an axonal terminal. What we're going to see is the electrical wind's going to come here, trigger open calcium gates, and those calcium gates are going to trigger a series of events. Right now, I'd like you to just step back and take a look at this. Here's your axonal terminal. This is known as the presynaptic cleft. Where's the word presynaptic come from? This cleft right here that's filled with gel is known as the synaptic cleft. That prior to it is presynaptic, that post to it is postsynaptic. Here's the muscle down here. Notice that this cleft is heavily folded, invaginated. Why? More surface area for these events to play out. Notice here on the postsynaptic, we're going to find a series of receptors, protein receptors that can regulate. Now let's take it a step further and look a little closer. Here's that same synaptic cleft. Here's the axon. Here's the muscle. This is called a neuromuscular junction. For that reason, it's a junction. It's a gap between a nerve and a muscle. Now here at this axonal terminal of the neuron, electrical event comes traveling down. That electrical event comes in contact with this set of proteins here. They're scattered throughout the axonal terminal. And what these are voltage regulated calcium gates. And these voltage regulated calcium gates, when the electrical event comes down, it triggers them open. Just so happens there's a whole lot more calcium on the extracellular space than there is in here. There's basically no calcium in here at rest if we've done our job right. So when those gates open, calcium comes flooding in. That's going to make the inside of this axon negative or less negative, basically depolarizing it. Bringing in those positively charged ions are going to stimulate these vesicles. And notice you got a whole flood of these vesicles here in the axonal terminal. These vesicles are known as synaptic vesicles, and they will house within them a neurotransmitter known as acetylcholine. And when calcium comes flooding in, it'll trigger these synaptic vesicles to migrate to the axonal terminal end, the presynaptic end, fuse with the presynaptic end, and through exocytosis release the acetylcholine into the synaptic vesicle. See this event played out here and here as well, the migration that occurs. I would ask you a quick question here. How come all the mitochondria are here? Well, when these gates finally do close, this electrical event ceases and these gates close, we're going to have to pump this calcium back out. And anytime you use the word pump, that means you're using energy. So the calcium is going to have to be actively pumped out, active transport. Let's get back to this. Acetylcholine gets placed into the synaptic clefts. Acetylcholine migrates across the synaptic cleft and binds to receptors that are chemical specific receptors here on the surface of the muscle. That would be the postsynaptic end. They are actually specifically there to bind to proteins that are sodium-potassium gates. When the acetylcholine binds, it opens the gates. Sodium moves. Remember, sodium, a higher concentration on the extracellular space than it is on the inside, so sodium moves in. The reverse happens to potassium. Just so happens, though, more sodium moves in than potassium moves out. And this gate stays open until one of two events occurs. Either acetylcholine stops being released into the synaptic cleft, or an enzyme actively digests this acetylcholine in space. That enzyme is known as acetylcholinesterase. Now, this creates a localized depolarization because remember, sodium migrates into an area that's heavily negative, and that causes this area to become less negative. Depolarization, a localized depolarization. Now, hopefully that localized depolarization, though, what will happen is that if enough sodium comes into this space, remember sodium ions are positively charged, what happens if we put two magnets together and try to position their positive charges against one another? They repel each other, right? Well, that's what we're going to see happening here. As sodium gets into this area, more sodium accumulates, it's going to repel and push outward. Well, as it pushes outward, it pushes out to these channels right here that are on the surface. Remember what these channels are? These channels are sodium gates, but in this case, they're voltage-regulated sodium gates. And so as the sodium moves out, are they going to bring a depolarized state in, a more positive environment, make this environment more positive? The answer is yes. When that happens and they change the inside of the cell to a more positive environment, it's going to trigger these gates to open, more sodium to come out in. As sodium comes in, it's going to migrate out, it's going to stimulate more gates to open, more gates to open, and ultimately what we see is a localized depolarization spread in what we would call an action potential. And that action potential then allows for a wave of electrical events to occur down the surface of the muscle. Here you see that kind of depolarization in numbers. At resting state, we're about 90-95% depolarized. Localized depolarization can result in changing the charge here. Right here is where we'd start seeing those sodium gates close, but they close slowly, and while they do that, they make the inside of the cell more and more positive. As that sodium comes in again, it pushes outward because it repels each other, the individual ions do, and we happen to have those voltage regulated sodium gates in the perimeter, which will open when that sodium comes out and causes depolarization. More comes in, we get a cascading effect that's called that action potential, much like we had in the nerve. Now, at this point, I'd like you to back up, take a deep breath like I'm going to, take a look at your muscle anatomy lecture notes, your sliding filament notes, and your excitation reactions, and get prepared for the way in which we tie all this together.